All right, we might make a start. I'm delighted to have been asked to introduce today's speaker at the Monday Lunch Live series, Professor Mark Dawson. I'm sure most of you here will know Mark, but I'd like, if I take a minute to describe some of Mark's underachievements over the last years. He's a clinician scientist here at the Peter MacCallum Cancer Centre. He's the program head of translational haematology, group leader, cancer epigenetics, consultant haematologist, physician, and also the Sandbrook Prize winner. And the work he's going to talk about today is largely the basis on which he was awarded that prize. Mark was awarded the General Sir John Monash Fellowship and Cambridge Commonwealth Trust Fellowship to complete his PhD at Cambridge. He was then awarded the inaugural Wellcome Trust Bade Prize, is that correct? To continue his work in AML and brand-domain proteins. He's a Senior Research Fellow for the Leukemia Foundation of Australia and a Howard Hughes Institute International Research Scholar. The title of his talk today is Can Epigenetic Editing Help Us Change Cancer Stories? Mark Dawson.
Thank you, Paul. So I wanted to just start off by saying how proud and privileged I feel to have won the Sandbrook Prize. Many of you may know Joe and recognise his amazing achievements over the many decades. It's unquestionable that he is certainly one of the pioneers of molecular biology. So to have anything associated with his name is a great privilege and honour. And so what I'm going to do today is just talk a little bit about our field of research and what kind of questions the lab is interested in. Talk to you a little bit about the prize and what that was given for, what body of work that was given for, and then show you what we've done since. So let's start. If I can get this to work. Have I done something wrong? There you go. Okay. So let's start with the clinical problem first. The lab primarily works on acute myeloid leukemia. And so it's worthwhile introducing this disease. Every year, about 900 Australians, but about 90,000 people around the world, I mean, we don't have accurate numbers, but it's around that vicinity, get diagnosed with this disease. It's characterized by abnormal gene expression and a block in cellular differentiation. And this leads to increased self-renewal of the leukemia-initiating cells. When patients like this come to see me, and I've got a clinic today to see patients with acute myeloid leukemia, it's a medical emergency. You don't really have a lot of time to procrastinate about what the best treatment option is. You need to get on and treat them fairly quickly. Untreated, life expectancy is measured in weeks to maybe a month or so. So therein lies the problem, because the treatment for this disease has really not changed in nearly 4 decades. The way we treated this disease in the 70s is the way we treat it today. And that's not because we have excellent treatments. So here are some survival curves for patients, newly diagnosed patients with acute myeloid leukemia treated with standard chemotherapy, which we still use today. An ideal curve would look very much like this, a straight line across the 100% mark. And clearly what you recognize from these curves is that we cure maybe 1 in 4 to 1 in 5 patients, which, gives you an idea of what the task at hand is. It's an expensive treatment and it is very aggressive chemotherapy. By and large, patients spend, a month each time in hospital during their treatment course. Now, just because we don't cure 3/4 of these patients doesn't mean we don't get them into remission. And this is a difficult concept for our patients to understand, and I go through this all the time with our patients. Cure is largely, remission is your first step towards cure, and cure by and large is identified with time. So how do we do with regards to getting patients into remission? We get about 80% of patients with standard chemotherapy into remission, but it's not sustained. And for those patients who relapse post their first remission, their prognosis is incredibly grim. It's essentially an incurable disease over the age of 60, And in a small proportion of these patients, we're able to salvage them with allogeneic bone marrow transplants. Now, the reason to go through this clinical scenario is to provide a context as to why there is an urgent medical need to understand this disease better and to find better treatments. What do we know about the disease? Hematopoiesis, like many other tissues, has been extensively studied. And like any other tissue, it's organized in a hierarchical structure. So at its apex are the small number of self-renewing hematopoietic stem cells. These cells have two unique properties. They're able to renew themselves, called self-renewal, and they're able to orchestrate organized differentiation for all of the differentiated blood cells that you know of, red blood cells, white blood cells, platelets, etc. What we think happens in acute myeloid leukemia, and this is a gross oversimplification, but it's worthwhile to tell the story, is that a leukemogenic event happens that either co-opts the self-renewal capacity of the hematopoietic stem cell or reinstates it to one of these progenitor cells. But then it has a block in differentiation, and therefore you get uncontrolled cell growth where the tumor arises. Now, leukemia is also organized in a hierarchical structure. At its apex are the small number of leukemia-initiating cells, or for want of a better word, leukemia stem cells. But the majority of the tumor actually is incapable of renewing itself. And these are the bulk blast leukemic cells. They're just not able to differentiate. What chemotherapy does extremely well is eradicate the bulk leukemic cells. But in 3/4 of the patients, it has no effect on the leukemia-initiating cells. So it's like pruning a weed in your garden. You pull it out and unfortunately don't get the roots. You feel better about it for a little while, but eventually it comes back. And that's what happens here. So you get relapse because you're not eradicating the leukemia cells. Why is chromatin biology or epigenetics important? Leukemia, unlike melanoma, lung cancer, or many other cancers, aren't riddled with many thousands of mutations in their genome. Every cancer is driven by a mutation in its genome, but in leukemia, this can be as few as just one translocation, or as many as just 5 or 6 mutations. So it's not a disease that's driven by ongoing mutations. What it's driven by is abnormal gene expression, or for want of a better word, transcriptional dysregulation. And we know this now because we've sequenced multiple genomes around the world over the last 5 to 7 years. And what we found is a central theme in cancer biology in general, but more importantly, this is especially prevalent in the myeloid malignancies like AML. What we see is that there are mutations in all of the protagonists that control the way our genome is accessed for either gene expression, for DNA repair, or for DNA replication. And by and large, these are called epigenetic regulators because they control the access to the genome, which is your genetic material. So targeting the epigenome, this has become an area of great focus for the pharmaceutical industry, for academia, and for clinicians as well. And whilst we used to have maybe 1 or 2 drugs that manipulated the epigenome to some extent, we now have a large compendium of these. And this has been, growing because of advances in medicinal chemistry as well as interest in the area. And this is what we have to do, what we have at our disposal today. What's highlighted here in red are new small molecule inhibitors that have now gone past the preclinical stage and are now being tested in the clinical arena in either early stage clinical trials or are now transitioning into the routine clinical practice. And we've had some part to do in this process, and we were involved in the early identification as BRD4 and BRD2 and 3 being really important regulators of transcription in acute myeloid leukemia. And this paper, which was published now nearly 6 years ago, was one of the first papers to show a new therapeutic paradigm for the treatment of aggressive malignancies. What we used here was we used a drug called a bromodomain inhibitor, and this drug binds and stops this protein here, BRD4, from physically associating with the histones, which is around which DNA is wrapped. And by doing so, and by displacing BRD4 away from chromatin template, it turns off gene expression that are driving leukemia cells. And therefore, these leukemia cells succumb to this inhibitor. Now, this was also shown in other hematological malignancies, including lymphoma and myeloma. And largely, these 4 initial studies, these preclinical studies, set the platform for the clinical trials that then ensued. What I can tell you is that we had no idea when we did this research in 2000. I think I started this in 2009 and it was published in 2011. Just how important this protein would become for transcriptional dysregulation, not just in AML, but in a range of both malignant as well as non-malignant hematological diseases. These are now, there's over 30 odd clinical trials that are testing these drugs around the world. And one of the privileges of being a clinician scientist is that not only was I able to be involved in the preclinical development of these drugs, but now I actually get to help run the clinical trial as well. And one of these clinical trials, this one here, is being run out of Peter Mac, amongst other centers around the world. Now, what prompted the study that ultimately led to the Sandberg Prize was the recognition of, both emerging clinical data as well as our background clinical knowledge. So the first early phase clinical trials from these patients are starting to emerge. And this is a different BET inhibitor, but by and large works exactly the same way. And what you can see here is that this is relapsed refractory acute myeloid leukemia by and large. So I showed you what those survival curves look like. Now, in some of these patients, even with such a grim prognosis, you do see single agent clinical activity. So the way you can look at this is two ways. One, you could say that the drug's not very good by itself, and you would have to agree with that, okay? It is not a panacea by itself. The second way to look at it is that it leads to a meaningful response in about 10 to 15% of patients, but we don't know why. Why is it those patients that derive benefit, and what is it about the others that do not? And so it says that we actually have to study this in far more detail than what we have done so far. The other thing we knew, which was inevitable, was that resistance to targeted therapies will emerge. We knew this from our clinical knowledge with probably what will remain one of the best targeted therapies of our era, which is imatinib or Gleevec. Completely revolutionized the treatment of chronic myeloid leukemia, but doesn't cure the disease in the majority of patients. We also see this in solid malignancies and other hematological cancers. The difference between the drug we had developed and all of these is that these are kinase inhibitors. And by and large, the paradigm for how resistance develops to targeted therapies was driven on what we knew about catalytic inhibitors. What we had helped develop was a non-catalytic inhibitor that disrupted a protein-protein interaction. So we needed to learn how this new, how will cancer evolve resistance to this new type of therapy. And so this is Chun Fong, who's a really talented hematologist and who started his PhD with me in Cambridge, and we started this project together there, and then he transitioned down to Melbourne with me and now leads the acute myeloid leukemia clinical service down at the Austin Hospital. And this is research that he did during his PhD. So what Chun did was he took primary hematopoietic cells that were taken from the bone marrow of mice, because we wanted to do this in a way that replicated the human disease. And we transformed it with a single oncogene. This oncogene called MLL-AF9 is sufficient by itself to lead to full-blown leukemia. It doesn't need any other mutations. Okay. Why did we choose this model? This was the model we actually established the efficacy of the drug in. So we wanted to model resistance in the same model. And then we took this model and we grew the cells as single-cell cultures in semi-solid media in the IC40 of the drug for about 2 months. And this was critical to getting this process to work. Now, each colony from these cells, from these dishes, is derived from a single cell, and we had mixed male and female bone marrow from the start so we could always know whether we were dealing with single-cell clones. We then took out these single-cell clones and grew them up in liquid culture, and then stepped up therapeutic pressure until we got cells that were growing well beyond the IC90 of the parental cells. The reason for multiple cells, single cell things, is what we wanted to control for artifact, that arose out of the culture process. And we wanted to keep the parental cells growing at the same time, and we wanted a vehicle-treated cohort as well, so that any kind of artifact that may occur was accounted for. So did we establish resistance? The answer is yes. So here are the cells now. They're all derived from the same original population. These are our resistant cells in brown. They're growing just as well as our sensitive cells. But when we challenge our sensitive cells, these blue cells, with the same concentration that our resistant cells don't care about, we kill them immediately. So chemical resistance had been established in this model. One of the other paradigms that we have in clinical practice is that when you become resistant to one type of targeted therapy, use it to be brother or sister. Right? An example of this is imatinib again, where you move on to nilotinib or dasatinib or one of the other nibs. Right? And we wanted to then ask, is that true for this type of therapy too, this epigenetic therapy? Could we just transition to a different chemical drug that does exactly the same thing, which is inhibit the bromide-domain inhibitors? And the answer was no. So if you were resistant to one chemical class of bromide-domain inhibitor, you are resistant to a completely different chemical class of bromide-domain inhibitor. We also went on to show that you are genetically resistant as well. You can knock down the gene using RNAi, and actually the cells didn't care about it too much. Now, not only could we show this in vitro in cell culture, but we can actually put it back in the animal. And here's the sensitive cell population, again, with this big survival benefit. I can tell you this is one of the most aggressive murine models of leukemia, so the survival benefit is indeed impressive. Or when we put our resistant cells back in, this survival benefit is completely gone. How does resistance develop? So we'd established the fact that we had generated a resistance model, but now we want to understand how it actually develops. One of the first things Chun noticed was when he was doing flow cytometry was that the physical properties of the sensitive and resistant cells were quite different. So this is a forward scatter and side scatter plot, which largely measures the size and granularity of the cells. And what you can see here in the red cells are these are myelomonocytic cells that are sensitive to the drug, but the blue resistant cells were much smaller and much more homogeneous as a population, suggesting they might be much more immature. So this is what we then asked, are these more immature cells? And in fact, the markers of myeloid differentiation that were present on our leukemic blasts are no longer present on our resistant cells, leading us to believe, could these be leukemia initiating cells? You know, these much more primitive cells from which leukemia develops. And so we went on to show that that was indeed the case in vitro, because you had greater clonogenic capacity from our resistant cells. But you know, the real test of whether these are leukemia stem cells is the ability to recapitulate the leukemia in a susceptible host. And how many cells did you require to do this? And so we did these limiting dilution assays and showed that our resistant cells were up to 1,000-fold more enriched for leukemia stem cells. Now at this point, we were cognizant of the fact that we developed a model of resistance that was completely artifactual to how resistance actually develops in a human being. You know, resistance, we developed a model of in vitro resistance. Did this have any relevance to how resistance should develop? So we went back and we redid a new model whereby we treated mice continuously until that therapeutic effect was lost, okay? Because resistance in human beings develops under sustained ongoing systemic pressure from the drug. And what we found is that everything we had uncovered in vitro also applied in this model, which is that resistance again emerged from the leukemia stem cell compartment. And here you can see that in our resistant cells, just 1 in 6 cells was sufficient to give rise to the leukemia. So does this mean that all leukemia stem cells are resistant to this type of drug? The answer is no. So if you take out a pure population of leukemia stem cells and you challenge them with sufficient drug that kills the blast cells, you kill off about 2/3 of these leukemia stem cells, as shown in this graph here. Okay. So it means that some leukemia stem cells are susceptible, but others are resistant. And we don't really understand this concept, and that's something that's, you know, it ties into the concept of intratumor heterogeneity. Just because cells look the same and behave the same doesn't mean they will respond the same to therapeutic pressure. And so that's ongoing work in the lab that I won't talk to you about anymore today. We also then went on to show that actually resistance takes time to develop. It's not that, you know, that it develops straight away. And this also is consistent with the fact that it emerges as a result of an adaptive process from the leukemia stem cell compartment. How does it develop? I told you that previously every paradigm that had evaluated a targeted therapy had shown that resistance largely developed by genetic evolution. That is, the cancer cell had another mutation that rendered the cell still competent, but the drug redundant. And so the cells were still able to continue on because this new mutation allowed them to. In fact, in this model, there was no single mutation across the various clones that we had developed that tied the story together. There was no mutations that allowed the drug not to bind, but the protein to still function, so-called gatekeeper mutation. So there was no mutational signature, no mutations in BRD2, 3, or 4, or no clear suggestion of causation. Now, this is an epigenetic drug, so was it still doing its job? Was it still keeping BRD4 off chromatin? And actually, when we looked at this, and so this is the amount of BRD4 that's bound to chromatin. You can see in the sensitive green cells here that there's quite a lot bound, and then our resistant cells is actually much less bound, okay? Which means the drug's still doing its job. It's still keeping BRD4 off chromatin. It's just that the cells don't seem to care anymore. Then we looked at some of the target genes of BRD4, including MYC. And we saw this, which is the drug's still doing its job because here's BRD4, and here's our resistant cells. This is sensitive and resistant. There's far less bound at chromatin. The interesting thing about this is that this gene MYC, which is very important for cancer cells, was expressed exactly at the same level in our sensitive and resistant cells. We looked at this at a global level and asked, well, what are the compensatory pathways that the resistant cells use to now overcome this therapeutic challenge? And one of the major pathways was this pathway here, the Wnt beta-catenin pathway. This was upregulated by our leukemia cells. Why was this finding important? Because it had previously been shown that in acute myeloid leukemia cells, the leukemia stem cells or leukemia-initiating cells had much more expression of this signaling pathway, Wnt beta-catenin. So it reinforced what we had found functional, that resistance was coming from the leukemia stem cells. We tested this, as we should do in science, both genetically and pharmacologically by manipulating the pathway. So here we took out a negative regulator of the pathway and showed that actually resistance developed within a week. It took Chun about 10 months to develop resistance in this model. But if you overactivate the Wnt beta-catenin pathway genetically, you can get resistant cells within a week, really tying into the fact that we had found the right pathway. If you downregulate the pathway, you reconferred sensitivity, you made these cells differentiate, they became susceptible to cell cycle arrest and apoptosis, and you reinstated the survival advantage as well. And importantly, we were able to show what happens at chromatin. How does this all work? So remember this gene MYC, where BRD4 is now bound in the sensitive cells, not in the resistant cells. If we look what happens, beta-catenin, which drives the Wnt beta-catenin pathway, in our sensitive cells, where BRD4 is bound, there's no beta-catenin. It loads up into our resistant cells to sustain the expression of these genes, and then it's gone again when we manipulate the pathway and make the cells sensitive again. And finally, the bottom graph here shows that actually the expression of the Wnt beta-catenin pathway in primary human patient samples with acute myeloid leukemia also predicted whether the patient would respond or not to the BET-broma domain inhibitor. So this is a summary of the work. The work was published 2 years ago, and I think there were some major take-home messages that I want to reinforce in the summary slide. The first thing is that we are used to analyzing cancer cell populations as a bulk heterogeneous population. And when you do this with acute myeloid leukemia, the red and blue cells here, which are the leukemia stem cells, are a minority of this population. In the majority of the population, the blasts, BRD4 drives their survival signature, their survival gene expression signature. So when we use a BET-broma domain inhibitor, we kill off the leukemic blasts and some of the leukemia stem cells, but not others. Now, genetics doesn't play a role in the evolutionary process here. What happens is that the cells adapt, and they're most plastic in the leukemia stem cell phenotype, whereby they say that program is no longer able to be used. I will use a different gene expression program to sustain the survival of the cells. And in acute myeloid leukemia, in this model, it appears to be beta-catenin that does this. This potentially brings up a therapeutic opportunity of targeting both at the same time, and we can talk during question time whether we think that this is a good or a bad idea. And I'll tell you now that I think that this is a bad idea. But we can talk about that in detail later on. So this is where that project ended. But I want to go back to the clinical findings, which is that, you know, that the drug works, but only in 10 to 15% of patients. It's not sustained. So really, it suggested that combination chemotherapies might be the next best way forward. And the issue with combination therapies is largely due to clinical need, which is you're faced with a patient in an urgent clinical situation and you've got to come up with a new therapy. This decision-making process clinically is driven by empiricism. And sometimes this works out okay, you know, when you're dealing with predictable signaling pathways. I'm going to show you some examples where it hasn't worked out okay. And remember that epigenetics is entirely context dependent. You know, an important aspect to remember is that every single cell in your body has the same genome, but it has a very different job description. Right? And that's primarily because your epigenome tells your genome what needs to be expressed at the right place, right time. So therefore, empirical combinations can sometimes be dangerous. So here's an example of a disease called myelodysplasia, where a drug that by and large leads to hypomethylation, which is a chemical modification late on DNA of the genome by this enzyme here, DNMT123. This drug inhibits this enzyme and actually has a profound effect in about 50% of our patients. You know, some of our patients can stay on this drug for 24 to 36 months, 2 to 3 years. You know, when I showed you that their survival benefit was so poor in the context of relapsed refractory leukemia. Now, here's another drug called an HDAC inhibitor that inhibits the removal of histone acetylation. Now, this drug has also been shown to have a modicum of response in myelodysplasia. So a number of clinicians in a number of sites around the world embarked on combination trials combining these two agents with the assumption that 1 + 1 should equal 5. Unfortunately, what they found, and this has been shown over and over and over again now, is that actually not only does it not lead to any greater benefit, but some of the patients who would have been predicted to actually do well with these drugs did far worse on the combination. So there's functional antagonism. And this really cautions against empirical use of epigenetic therapies, because this is what we now have to play with. And as I said to you, many of these are now transitioning into the clinical arena. So Omar Gillen and Enid Lamb in the lab tried to work out the molecular rationale for why we would use 2 epigenetic therapies. And this is a paper that was now published a year ago. And what they were able to show in this paper, again, in MLL fusion leukemias, where it had previously been shown that an histone enzyme called DOT1L was very important for the survival of these cells, and we and others had shown that BRD4 was important for the survival of these cells. They wanted to work out how they worked. Did they work in completely independent fashions, or did they work together? And the answer is they actually work together, but as separate complexes. What happens is this histone enzyme first comes along, and it puts down chemical modifications on histones. It methylates a specific residue called lysine 79 on histone H3. This doesn't change the gene expression of the cells, but what it does do is that it relaxes the chromatin architecture, making it more permissive for transcription factors to come and bind. And these transcription factors then bring with it another histone enzyme called a histone acetyltransferase, and this then acetylates histones, and it is to this that BRD4 then binds to drive the gene expression that sustains the leukemia cells. Why was this molecular mechanism important? Well, we now know exactly how the gene expression program in these cells is set up. And while we may not be able to drug both without causing excessive toxicity, we have a number of other avenues where we can build different drugs. Okay? And that process is very important. I want to now tell you about a brand new story, that is completely unpublished, and talk to you about why we embarked on this. One of the things that became apparent to us as we were doing our research is that we're really blinded to how these drugs work. You know, you put them on cells or you treat animals with it, but you really don't have any idea where in the cell the drug is or what is it actually engaging in the cell, etc. So we asked a set of defined questions about if we were to build an ideal drug and do molecular biology research with small molecules preclinically, what would be the ideal skill set to have? From a mechanistic point of view, we'd want to know where in the cell it is. Okay? We'd want to know what its molecular target is. What's the molecular target of the drug when it's binding its protein of interest? And what else is in the vicinity? Because we work with drugs that act at the epigenome, we know that this is not stochastic. Okay? We know that when you put a BET-broma domain inhibitor on, you get about 400 genes change their expression. Why 400 and not several thousand? You know, what's special about those genes? What about in a whole organism? You know, every clinical therapy that gets to the clinic usually goes to an animal model to assess the efficacy of the drug in that animal model. But again, in an animal model, we don't really know what cells the drug gets into. Does it affect cancer stem cells? Does it affect normal cells? You know, so these are the questions we set out to do about 4 years ago. We did it in collaboration, and this could not have been done if it wasn't done in collaboration with some really special people who have very great skill sets that we don't have. So Paolo Grandi, who works out in Heidelberg in Germany, they specialize in quantitative mass spectrometry and do, you know, proteomics at a level that is really not done at very many places in the world. Rafael Rodriguez is one of the best medicinal chemists I know. He works out of the Institute Curie and really is an innovative thinker in terms of how he thinks about problem solving with chemistry. And Rab Pringe is the head of the academic division of GSK that works on epigenetics, and his research group are interested in structural biology and how, you know, how proteins form certain structures and how best to drug these structures, and also does quite a lot of medicinal chemistry. This work was done by Dean Tyler, who started his honours with me and has now finished. He's finishing his PhD. He hasn't finished yet. He's well on his way, and you know, we're pleased to say that this work actually is Dean Tyler. This is not normal time for Dean to turn up to work. He's been here for a while. So Dean, you almost staged that. Did you know I was going to we're really pleased because this work that I'm about to tell you about was just accepted for publication in Science. So let's tell you how we did this. So we used something that had previously been invented, a method called click chemistry. And here what you do is you attach onto your small molecule an inert functional group that doesn't react in the cell, but provides the ability for you to be able to click on whatever else you might want to do, be it a fluorochrome, an epitope tag, etc., etc. And when this click reaction happens, it's stable. And it's a covalent reaction that cannot be reversed. You can do it several different ways, and we use the two most common, which is by metal catalysis using copper, or in a copper-free dependent way, because as you can imagine, depending on what question you're asking, you might want to do one or the other. And using drugs as a scaffold to be able to ask some of these questions, you know, which is what proteins do the drugs engage, etc. Well, that wasn't new. You know, in fact, we had done that. Back when we started to work on BET-broma domain inhibitors, it was how we found that the BET-broma domain proteins actually engage with transcriptional complexes. Okay? Other people have used drugs like this to localize the drug on the genome of cells. The issue we had is that these bulky drugs, they're good for certain in vitro questions, but they actually don't get into the cell. Okay? And they don't phenocopy the molecular activity of the parental compound. Here's the evidence for that. Okay, so here's this drug. Well, it's about as effective at killing these cells as the vehicle is, as the MSO is. Okay, but here's our clickable drug, which is the molecule highlighted in red here. It kills the cells identically to the parental compound. And also we see this at the gene expression level. I'll go through a few more of these details. So every single phenotype that we had previously established for a bet-bromodomain inhibitor, we read it again with our clickable compounds. And in every single assay, we were able to show that the drugs we had developed completely phenocopied the effects of the parent compound, be it in proliferation, viability, the induction of apoptosis, cell cycle arrest. When we used it in cells to go and engage the proteins at the chromatin interface and displace them off chromatin, it worked exactly the same way. And probably the best example of this that gives you, well, gave me unequivocal confidence that we had developed drugs that were identical is this result. So here is, we were analyzing the global transcriptome, so every single gene that's expressed and turned on or off in a cell following challenge with a bet-bromodomain inhibitor. The correlation between our clickable compound with our parent compound was 98%. That's the equivalent of just doing the same test over and over again with the parent compound. So we've now developed drugs that were clickable, that were able to phenocopy the parent compound. What could we do with these drugs? The first thing we did was we were able to go and localize where in the genome these drugs bind. Okay, so here is where BRD4 is bound in the genome, and this is where our drug is bound. If we zoom into the gene, which I've been telling you about, called MYC, here you can see that BRD4, our target, is bound in green here, and our drug is bound in purple right here. Because these drugs are small and get into the cell so easily, we can elute in non-clickable drug to titrate away any background that may occur. So we can very specifically identify exactly where in the genome the drug goes and binds to it. Why is this important? Because the way the drug works is not really fully established. Okay? When BRD4 is bound at the transcriptional start site of genes, it's less susceptible to being displaced by a bet-bromodomain inhibitor than when it's bound at enhancer elements. So what localizes and keeps it there are two different things. And just the amount of protein that's bound there is not very good at predicting this, okay? Because it's exactly the same in the genes that go down with the drug than those that go up. What does change is the environment, the histone acetylation around the genes that respond or not. And we had made the assumption that the amount of drug that gets to the gene may actually predict whether the gene goes down or not. And this is exactly what we see. So now, by localizing how much drug goes to a particular gene, you can tell whether we're likely to turn that gene on or off, and you can do that in a single test. Okay. How does it work? How are we able to actually localize the drug at chromatin? Our hypothesis is that BRD4 uses one of its bromodomains on histones, and the drug fits into the other pocket. But we had to show that. And so the first thing Dean did, or one of the first things we did to try and show this, is that we took cells that we knew would die as a result of knocking down BRD4, and we rescued these cells with either wild type or mutations in either the first or second bromodomain. And what we see here is that the wild type protein rescues the cells. The mutation in the second bromodomain rescues the cells by and large, but it's the first bromodomain that is really important. Okay, so it tells you that this is the domain of the protein that helps localize it to the chromatin template. We're also able to use small molecules to try and tease this out. So here are some drugs that have different potencies. And what we were able to show is that drugs that have a higher potency for the second bromodomain here, RVX208, well, this actually doesn't displace the protein of interest away from the chromatin template at all. And here is the amount that is bound at chromatin. RVX does not pull any off into the soluble fraction, whereas the drugs that kill the cells, which are these two, as you can see, there is more protein coming off. Finally, the proof of that, this is how we're localizing it to the genome, is shown in this assay here. What we were able to show here is that in using the analysis that we had just developed, here BRD4 is localized by an antibody detecting the BRD4 protein at the chromatin template called CHIP PCR, and then by using the drug to actually localize it at the same place called click PCR, we see that it's bound at the same spot. Now, what a bet bromodomain inhibitor does is it binds both bromodomains and displaces it away from chromatin altogether, and therefore you get loss of both the chip and the click signal. By using a drug that specifically occupies only the second bromodomain, you reduce very selectively just the click signal. So we also see, we then went on to look at what happens at enhancer elements. And in enhancer elements, we also see this process where there's actually quite a lot of drug at some enhancers, but not a lot at the other. In contrast to what happens at the gene promoter, here they both require the first bromodomain to be bound. Okay, so what we think is happening at these genes is that a transcription factor of something else is blocking access to that second bromodomain. We were able to show that that transcription factor is a transcription factor called CBP alpha, which is really important for myeloid cells, and show that it is localized exactly where the ID4 is bound. Why are these tests important? I mean, we've gone into quite a lot of granularity, but actually what we were able to show here is how this protein binds in selective genes within the genome, and we would not have been able to do that without manipulating these drugs to be able to be used much like antibodies are. Okay. I told you before that, you know, sometimes people, when you become resistant to one type of chemical against a protein target, use a different drug or against the same target but a different chemical compound. And I'm often asked whether that's the case. You know, if you become resistant to one bromodomain inhibitor, you become resistant to the others. We've previously shown this, but people still ask. So what we decided to do was look to see if the two different chemotypes for the bromodomain inhibitors bound the same proteins in the cell. We did this using a process called click proteomics. So here we used the compound to bind the protein targets in the nuclear lysate of cells. And here when we capture these, some of them are bound in a very specific way, which is, as you can see here in the red, whereas others, because this has a charge, will bind the proteins in a non-specific way. To distinguish between this, we dilute in free drug, which then brings the very specific complexes of chromatin. And when we do this, we see that regardless of what chemical type you use, the bromodomain inhibitors associate with the same nuclear complexes to the same extent, which really gives us reasons for why one compound doesn't overcome the resistance to the other. Now we wanted to actually ask something different. You know, can we actually see the drugs, right, using these new molecules that we developed? I won't go through how we did this, but essentially now we can click on a fluorochrome and show you that here is the drug, here is its protein target, and it's completely co-localized in either solid malignancy cells or hematopoietic malignancy cells. We can also now quantitate how much drug is in a particular cell using flow cytometry. Okay? And this is a dose titration where we put in more and more drug. And this is very specific because if we put in a competition assay with drug that is not able to be clicked, you see an equimolar dilution of this. We next did an assay to ask, what about in a blinded fashion? If we were to actually grow some cells in a high concentration of drug, another in a low concentration, mix them together, can we find out which cells actually got more drug? And whilst the concentration of drug doesn't change the physical properties of the cells, we could clearly distinguish now which cells actually got more drug. This gave us the confidence to do the next analysis, which is now say, what about in a whole organism? Right? Can we actually treat a whole organism to find out where the drug goes and whether it affects cancer cells equally in different tissues? Why would we do this? One of the assumptions we make in clinical trials and clinical practice is that the chemotherapy works just as well regardless of where your tumor is located. By this I mean if you have tumor in your liver, spleen and bone marrow, the drug will work equally well in all of those tissue compartments. We know clinically actually that's just simply not true. Okay, and here's a mouse model that phenocopies what we see clinically, which is when this mouse has overwhelming leukemia and we treat it with a bromodomain inhibitor, we clear the leukemia from the peripheral blood, which is up here. We get rid of the leukemia from the spleen, but from the bone marrow, we largely don't touch the leukemia, okay? And this provides an example of why this leukemia potentially comes back. Now, we wanted to know why this might be the case. So we now dosed the animals with a clickable BET inhibitor and asked how much drug goes into these different compartments. The peripheral blood obviously gets the most amount because that's where it's first absorbed. But remarkably, we were able to actually show that much more drug gets into the spleen than it does into the bone marrow. We don't know exactly why. What we can tell you is what aren't the answers. Cell cycle, how quickly the cells are replicating, et cetera, has no bearing on this. And nor does the amount of actual target that's present in these compartments. But it's a very clear result. We know that this result is likely to reflect what we see clinically because the compound we use has many of the same chemical properties as the original compound. So this is a log p-value of the original compound, and this is the log p-value of the compound we used. We also went on to show this actually by actually now dosing the mouse with the parent compound, taking out the cells from the spleen and bone marrow, and quantitating how much drug was in those cells by quantitative intracellular mass spec. And we saw exactly what we saw with the click chemistry, which is there's far more drug in the leukemia cells in the spleen than in the bone marrow. We can also now use this to talk about, therapeutic window. It's something we talk about clinically all the time. Are we likely to affect cancer cells more than normal cells? The way we currently do this is in a dish, right? So we wanted to do it in an animal, where from the same tissue, we could take out normal and malignant blood cells and ask how much drug got into both of these compartments. And we were also able to show that the amount of drug that gets in directly correlates with how much the gene expression has changed in these two different compartments. Finally, one of the things we do all the time clinically is assess the tumor using histology and, to a large extent, immunohistochemistry. So here we were able to take a cross-section through a mouse femur. This mouse had a 70% leukemia burden. These leukemia cells express a green fluorescent protein, which is why you can see them. And this is our drug. Okay? So now we can look to see in a very granular way which cells have the drug. Is the drug mostly present intracellularly? Does it work in the tissue microenvironment? We can co-stain this with any other marker you want to find out how the drug is working. So that brings me to my summary slide on this. Why was this an important piece of work? I think it really changes the way we evaluate drugs in a preclinical setting. It allows us to really find out where in the cell the drugs are going. It gives us the opportunity to actually identify what the proteins are that the drug is targeting within the cell using proteomics. We know we can find out where in the genome the drug is acting. And really, for the first time, we can show in the whole organism, is there different tissue distribution? Does the drug preferentially access and eradicate cancer cells versus normal cells? So I'm going to stop there and take questions. And there's a lot of people to thank for this work. They're really highlighted here in red. So I told you about Trun's work, which led to the Sandberg Prize, Dean's work, which is the last part of my talk, Omar and Enid's work. The people in the lab who make it a fantastic environment in which to work in. I don't ever feel like I'm at work. I feel like I come to work to have fun. It's great. Even though we run two separate groups, both Sarah Jane and I run it as one large group. And this is a photo from a recent lab retreat where it was really comforting to see what a fantastic group of scientists we've assembled, and the fact that we've been able to answer some very important questions. Thank you.